Hi, I'm Shea. I'm going to be presenting my engineering senior design project, Photopatterning Life, Multicolor Optogenetic Control of Cellular Patterns, and I completed this project with my partner, Lucas Shaki, right here. So why do we care about cellular patterns in the first place? First, when I talk about cellular patterns, I'm referring to the shapes and arrangements of cells that really ensure their development and function. So if we look at an artery or a capillary, for example, you can see rings of different types of cells that allow them to function the way they do. Even comparing these two that have pretty similar ring shapes, you can see differences in cell types, differences in thicknesses that make them more suited to pump blood quickly or exchange nutrients. And these are actually some pretty simple shapes and geometries compared to the geometries in the human body. For example, our team is really interested in the lung, which has this really complex branching structure like a tree. And when you zoom in even closer, there's also a beehive-like structure that's really crucial to oxygen exchange. And for any of these geometries, if there's any defects or incorrect arrangements, it can really impair your ability to live. The problem is, even though we know the importance of these in cell function and in disease, we lack the tools to produce and investigate these complex patterns. So current tools available include seeding molds, like those shown here. However, these are actually owned by one company, patented technology, and they only produce the molds in these shapes, these rectangles or this wheel shape, which are clearly not biologically relevant geometries. We almost never see a pie shape in the body. New technology, including cell stamping, has tried to improve the geometries we can produce. This technology creates a physical stamp, and you kind of paint a cell adhesive like a glue onto it. You press that onto a surface you want cells to stick to. But if you want multiple types of cells, you have to produce multiple stamps and do sequential layers of cellular adhesive and line up these stamps perfectly. So this is really slow and resource intensive. So our project tried to take a different perspective. Instead of controlling cell placement, we're going to control cell activation. So we're going to turn on a region of cells next to another region to have different populations of cells in diverse patterns. To do that, we're using optogenetic split recombinase. So to break this down, optogenetic means the control of gene expression using light, and split recombinase is a type of enzyme that can cut specific sequences of DNA. So on the left, you can see the schematic of our setup. We have a high-powered projector called a digital light engine that projects light in any pattern we want onto a mirror that gets reflected up onto a plate of cells. And this is the image of our actual hardware setup in the lab. So to start off, I'm going to explain how these recombinase enzymes work. So for the purpose of this presentation, we're going to think of the recombinase enzyme as a pair of scissors, but not just any scissors, the fun craft scissors. So as I said, recombinase can only cut specific sequences. So for example, these red wavy scissors can only cut this red wavy sequence of DNA. Meanwhile, the jagged sequence can only cut the jagged sequence of DNA. And these bottom two strips are actual DNA, a representation of our DNA constructs we've used to control cell activation. They both end with a fluorescent gene, which is basically a gene that allows cells to glow. But in front of the fluorescent gene, we put a stop sequence. That means that we can't get to this glowing gene, and the cells don't turn on. But when the enzyme cuts its specific sequences, it cuts out this stop sequence, and we see the cells glow. So the way we've worked in optogenetic control, or control with light, is we've actually split the enzyme into two halves, like having two halves of a scissor. And if you've ever tried to cut a piece of paper with half a scissor, it doesn't really work. So as it stands, you can't cut up at that stop sequence, so you don't see the cells glow. But when we add a specific color of light to half the scissor, they come together and they can functionally cut DNA again. And they cut out this stop sequence and we see the cells glow. So using this tool, we can have spatiotemporal control over cell activation, which means we're controlling cell activation over space and time. So a cell that saw light would be activated, and a cell that did not see light is not activated. And we can use this to pattern a single cell, a group of cells at the same time, or even an entire well of millions of cells. The image you're seeing here is a well of over 1 million cells, and every green dot is a cell that's been activated by blue light. So what have we been able to achieve with this system? We can achieve really high contrast, high resolution patterning. We can pattern features down to 100 micrometer scale. And we've also been able to kind of have fun with this and produce some really complex patterns that you can't produce with existing technology, including this checkerboard, DNA helix, the BU logo, or a lung. And because we use a projector system as opposed to a microscope like many other systems have used in the past, it's highly scalable. So we can pattern a small well of a couple 100,000 cells or an entire plate of millions of cells very quickly. So this image is a Pac-Man board we've patterned into over 50 million cells in only 3 minutes. Also, because these recombinase enzymes only act on their specific sequences of DNA, we can layer these tools to have multicolor activation. So these patterns are controlled with both blue and red light input. The first one is a cell made of cells. It looks like an avocado, but it was meant to be a cell made of cells. A Tetris board, and we have a Pac-Man ghost to keep with our theme. And this is actually the first example of multicolor optogenetic patterning without a photomask. Using this system, we are also doing spatial computational logic. So using the pattern on the left, we could control, like a computer, the space where cells are activated. So if we take the AND gate, for example, cells would only activate if they see both red and blue light. Or with the OR gate, they would activate if they saw either red or blue light. So to go over the key advantages of this tool, we can achieve really high contrast, high resolution patterns from a variable scale, patterns on a micrometer scale all the way up to centimeters. And we can do all of these scales in 3 minutes of illumination. Also, using our custom MATLAB script, we can upload any image we want to our projector, so we can quickly iterate through patterns, which is really necessary for developmental systems. There are many near and far applications to this project. Our team is particularly invested in developmental models of tissue development. As I said before, we're interested in how the lung develops, so we're using these light inputs currently to try to model what early tissue development in the lung looks like. And we've also begun building a 3D bioprinter, and we envision being able to cross-link a material while also defining the cell fate as we print an organ. Thank you. Any questions?
Hi, phenomenal presentation. I was wondering, do you believe this could be like a living canvas for artists? And are there any people out there that are actually using this? Because these images are fascinating.
That's a cool idea. I was thinking about that with the Kilachand showcase, actually, like kind of making like a gallery of some of the cool images we've made. In terms of optogenetics, no one's currently using it to make art, but I know there's some professors that I really look up to that create like cellular art in general with different like stains and stuff. So I feel like this could be our into that world. Yeah.
Hi, Shea.
Hi.
I know you mentioned that the existing technique with the plastic is patented and that the other technique with glue and staining can be kind of labor intensive. I'm wondering, I'm not a scientist, but I understand research is quite expensive. I'm wondering how your innovation compares to the other two in terms of price, both in the short and the long term.
Especially in terms of the seeding molds, those are like a one-use thing. You would seed cells in, you remove it to be able to do any experiment, and then that's like contaminated. It's usually ripped because you can't really pull it off well. So it's definitely a cheaper initial cost, but you would buy those every time you wanted to run an experiment for each well, where this would be an initial investment because you would purchase the projector system. But once you have the projector system, you can pattern any scale over and over again. Lucas and I run like, I don't know how many cells, like at least like 100 million cells every week. And it's like no extra cost to how many times you do. So long term, this is more cost effective. Short term, maybe the other one.
Any other questions?
This is so great, Shea. What was the biggest thing that you had to kind of like troubleshoot or trial and error in developing this model?
There was a lot. I actually joined this project in my freshman year of college. It's come very far from then, but the thing that's really difficult about this project, but also really cool, is it's like really truly bioengineering in my opinion because it's like engineering DNA, engineering a cell, and engineering hardware. It's like all of the levels you think of. And so I think that was what was difficult because when something goes wrong, you don't know which level to troubleshoot first. Like, is it a DNA problem? Is it a cell problem? Like Lucas and I's conversations are usually we go through like a whole list of like, well, what about this? What about, and we have to like knock each one off in order to find finally what the problem is. So yeah, I would say like interaction between the like molecular components and the hardware optics, having to like modify them at the same time. You change one, it impacts the other. You change one, it impacts the other, and it just keeps going like that.
Amazing. Thank you, Shea.